Privilege of being the head of School of Natural Sciences here at the university, and I'm very proud to have Associate Professor Amy Cain as just an outstanding and super member of my school, so it's an absolute pleasure to welcome you here tonight to hear Amy speak to us. I'd like to start the proceedings by playing this short video to acknowledge the traditional owners of the lands on which we are on tonight.
Welcome. We acknowledge the traditional custodians of Macquarie University land, the Watamaragal clan of the Darug Nation, whose cultures and customs have nurtured this land since the Dreamtime. We pay our respects to elders past, present and future. We welcome people of all nations and all faiths. Kwai Bidja, Jamna Payala Janawi. Come here, we speak together. We have 60,000 years of archaeological evidence of Aboriginal habitation at Lake Mungo and 20,000 years in Ryde. We have great antiquity. Today, hundreds of Aboriginal and Torres Strait Islander people graduate from Macquarie University. The Darug Nation had famous leaders such as Chief Yarramundi, Naraginji, Colby and Maria Locke. Many of the descendants of these Darug people live today amongst you. We celebrate with you our ongoing attachment and custodianship of this country. We celebrate the achievements of Macquarie University.
So again, welcome, and it's my great pleasure to introduce tonight our speaker, Associate Professor Amy Cain. So if you to walk around our school, around our faculty, around our university, and you mention Amy Cain's name, what you will hear is that Amy Cain is just a superstar. There isn't much in her rather short career that she hasn't achieved, and I'm sure there isn't anything within her full career that she won't. She is indeed a superstar. Amy joined the university in 2018, having spent some time abroad in various institutions. So she completed a PhD at the University of Sydney in 2012. She moved over to the UK and worked in the Wellcome Sanger Institute, working on DNA and RNA sequencing. She then went to the Wellcome Trust Clinical Research Program with the University of Liverpool and worked in tropical medicine in Malawi. So she has fantastic stories of the work done there. She then came back to Macquarie for a while, went back to the UK, worked with Vertex Pharmaceuticals there. So she has this fantastic array of skills that span industry and pharma and research and genetics. And today we're going to hear about some of the fantastic work she's been doing. Much of her research focuses on developing new antibiotics and understanding antimicrobial resistance in pathogenic bacteria. But what Amy also does is a lot of work convincing bacteria and convincing other critters through modifying their genetics to do really, really cool stuff. And I'm sure we're going to hear a lot about that today. She's even got some show and tell over here that you'll be able to have a look at later. So I mentioned that Amy is a superstar. She has a list of awards and achievements that's about this, well, longer than this. That includes the David Sims Research Prize in 2023, and most recently, the very prestigious Gottschalk Medal from the Australian Academy of Science that recognises her work in the field of antimicrobial resistant bacteria. When she is not being a superstar of science, I am told that Amy is human and does have some fantastic pastimes. You will find Amy walking her dog, and as she described it to me, he's a cross between a corgi and a dachshund, which is like two sausages coming together. She's very fond of her dog, getting out on bushwalks, and also loves a spot of yoga. So, without further ado, please join me in welcoming Associate Professor Amy Cain, and please do enjoy tonight's presentation. Amy.
Thanks, Grant. That's one of the nicest introductions I've ever had. Ah! And I feel very exposed that you know I have a double sausage dog now. I just realized I should have put this on before, but I'm a pacer, so I'm going to put this on me. All right. So, I'm going to talk to you today about how microbial genetics can improve our world. I'd like to thank the organizers for not only inviting me to speak today, but also for putting on this amazing lecture series. There's a lot of faces even in this room that I see, and I, might have coffee with or whatnot, but actually don't know what they do with their science or research. So it's really fantastic that we can get together and really understand what other people are doing on campus. And I hope, there's a long question and answer session at the end. So if you have any questions, no stupid questions. You can ask anything you'd like about microbiology or genetics or whatever. So I thought I'd talk to you, start with how I got interested in microbiology. So my journey sort of started, sorry guys, I really should have done this beforehand. My journey started actually in Mexico. And what had happened was that I had just finished my undergrad in molecular biology and genetics at the University of Sydney, and I wasn't sure what I was going to do next. And I went to visit my friend who was studying in Guadalajara in Mexico, and just had a little bit of a break when I was deciding what project I was going to do or if I was even going to continue studying. So we saw some amazing ruins, we went to some amazing markets, and of course, we ate at some local cantinas. Now, this meant that we ended up a few days later in a hospital room in the middle of nowhere, and basically had a makeshift drip made from our own scarves, a coat hanger, and hanging off the fan, and some drips for a good week. Luckily, we had some doctors come visit us, and they took some blood, and the blood samples revealed that we contracted typhoid. Now, typhoid is caused by this guy here. This is Salmonella typhi. It's a bacterium that gets into your body, into your blood system, and it causes havoc, causes fever, diarrhea, vomiting, the whole shebang. So luckily, we did move into the hospital once they realized that this was quite serious, and we got some really potent antibiotics, ciprofloxacin, and a few days later, the antibiotics cleared up the infection, and we're able to continue on our travels down in the Yucatan. So that was all well and good, but ironically, the very day that I was going back into the hospital and I knew I had typhoid, I had to make the choice if I was going to continue studying and what I was going to study. So there was one project that was offered on Salmonella at the University of Sydney with Ruth Hall, and I was like, yes, I'm going to do that. I'm going to get my revenge on these guys. And I worked on that for quite a while after that. So this experience led me to think, like, what would have happened if, we'd been in the middle of, rural Mexico, and the antibiotics hadn't worked, and we'd, gotten sicker and sicker. And actually, this was probably a driving question for the rest of my career. What I realized is that antibiotics truly are rare miracle drugs. We don't have a lot of drugs on the market that are cheap, really effective, easily available, and you only need to take them a little, for a week or so with very little side effects, and they increase our, lifespan greatly. They treat a whole host of infections, from wound infections to STIs to lung infections. There's nothing that they can't do. So these have been around for almost 100 years, since Alexander Fleming did his famous, I'll recreate it for you, noticing that there's clearance zones. Actually, we do have clearance zones at our display table, showing that some fungi were killing these bacteria, and this is how he discovered penicillin. So this was in 1928. In his acceptance speech, about 15 days later, yes?
He discovered antibiotics because his lab was a mess.
Yes. Yes, he was famously messy, and he let the fungal spores, which are in the air, settle on the plates because he didn't wash them up. Yeah, someone that's quite messy myself, I agree with that approach. So basically this was over, in 1928 he discovered the antibiotics, and then when he gave his Nobel Prize speech about 10 years later, he noted even in the speech that although these are miracle drugs and they're really effective, it's not that hard to make microbes resistant to penicillin. So you can see this happening in the lab. And the sad thing is that this happens in the real world, and this is a huge global threat to human health, and it's on the increase. So our best guess our best estimates show that by 2050, there's going to be about 10 million deaths per year due to antimicrobial resistance, which is more than all cancer deaths combined. Okay, so this is a big problem, and it's getting bigger. I have to tip my hat to the bacteria, though, who are evading these antibiotics, because they do this in really clever ways. So the ways that they become resistant is either they can produce little Pac-Man enzymes that will eat up the antibiotic. They can change themselves their own targets so that the antibiotics don't work or they don't get in the cell. Or they can have little pumps that they can pump out the antibiotics as soon as they enter their cells. So they're really clever, and I have to say I'm a little bit, I respect them deeply. And the more I look into microbes and my experience as a microbiologist, the more that I realize that microbes really are talented, wise daredevils. In fact, their superpowers improve our everyday lives in probably more ways than you know. So, for example, here we have our baker's yeast, if you know this. So this guy is called yeast called Saccharomyces cerevisiae. There's a couple of yeast aficionados over there. This guy allows us to brew beer and bake bread and a whole bunch of things that you'll notice in your everyday lives. I've already mentioned this guy. This is penicillium. He's a fungi that produces antibiotics, but there's a whole bunch of fungi and other bacteria that produce our life-saving drugs that we use every day. And lastly, here's Pseudomonas. This guy grows on contaminated soils and oil spills and things like that, and we can use him for bioremediation or other cool stuff like that. In fact, these are just a handful of examples, but really microbes' talents are endless. There's no surface on Earth that is sterile, right? There's microbes in every type of environment that we've come across so far. So here we have microbes in sulfur vents. We see them in the Antarctic. We see them in acidic soils and contaminated land. We see them everywhere. And if you think about it, we can harness these powers if we can only understand how the microbes do these things. So how can we understand and harness these talents of microbes? Well, microbes are really hard to look at. Clues in the name. So if we take the kind of microbes that I like to study the most, which is bacteria, about 10 million bacterial cells can fit on the tip of a pinprick, right? So that's not going to do you much good by just looking at it. What we can do is grow them on plates like these. Now, these plates, or agar plates, are actually like millions of cells growing on top of each other in communities. They're not single cells, but we can get an idea of what's going on as an average of a community. The other thing we can do is look down a microscope. So here's an example of a really powerful microscope, an electron microscopy image of the bacteria Klebsiella pneumoniae. Here we have an intact cell, and then you can see some of the cells are being completely bust open, and this is because in this media there is a really powerful antibiotic called colistin, which likes to pop open bacteria. So that's a really good antibiotic. So that's all great, right? We can see a little bit to do with the bacteria, but it doesn't give us a lot of information about what's really going on. Plus, most microbes in the world, like 90% of them, can't actually be cultured. This means we can't really grow them in the lab. So that microscopy isn't really great for that. So what we can do is look at their DNA. The DNA is the building blocks of life and can give us like a lot of information about the microbes that we're dealing with. So I don't know how much you guys know about DNA, but I like to think about it like the writing in the cookbook of life. So bear with me. So with DNA, instead of having 26 letters like in our alphabet, there's only 4 letters, A, T, G, and C. And just like the alphabet, they don't make sense if they're sort of jumbled up or if they're on their own. They have to be arranged into specific words, which we call codons, and then sentences. And then ultimately these are arranged into chapters, which we call genes, right? So genes are the things that instruct the cell or are the instructions to make proteins. Now proteins are the kind of doing part, right? So we know there's keratin in our hair, that's a protein. We've got collagen in our skin, that's a protein. And there's single genes that are instructing the cells how to make those proteins. So overall, we have the whole book of life, which is around, for bacteria, 500 of these genes or chapters, and this gives you instructions on how to make the whole cell or the whole organism. So how do we look at the genomes? This is the total DNA in the cell or the book. We can sequence them. So genome sequencing is getting much faster, cheaper, and more effective. Back in 2003, when they first sequenced the human genome, it cost $3 billion and 15 years and a lot of people's blood, sweat, and tears, I can tell you, to get one human genome. Now, 2025, we can send it off in 2 weeks' time. You know, you can get it back, pay $500. In fact, a lot of the people in this room might even have either their whole genome or part of their genome sequence as part of 23andMe or Ancestry.com or something like that. So it's becoming really accessible and much more common. And this is largely due to the differences in the technology. So there's been an explosion in innovation in sequencing technologies. Back when they were doing the human genome in this sort of Sanger sequencing traditional model, basically they were using these large like agarose gels like you would make in the kitchen, like that are agar plates basically, or they're made of jelly. So they used to hire chefs to make these giant gels. They put DNA and run them down there, and the length of the DNA would basically tell you what the order of the bases was. So you could order the bases that way. Obviously that takes a long time and is really laborious. Now we have these next generation sequencing technologies. Here I'm playing with a Illumina machine. This one is really one of the things that accelerated this sort of sequencing. And funnily enough, both of these technologies were invented pretty much by one guy. This guy was really smart. His name's Fred Sanger, and he won, but not one, but 2 Nobel Prizes for his work in genome sequencing technologies, but also in sequencing and understanding the insulin gene and protein. So since then as well, I mean, there's other technologies out there like these MinION or like nanopore technologies, Prometheans, you might have heard of these. These are actually the kind of really portable, you can see I'm holding one in my hand. These just plug into your laptop with a USB, and you can start getting sequencing information after 30 minutes. So you can use these for like outbreaks in the field. They're much more portable and they don't cost as much. So sequencing is getting heaps more accessible. This means that we're absolutely drowning in genomic data at the moment. We're getting heaps and heaps of genomic data. So back in 2003, when they were sequencing the human genome, there was 26,000 genes available, like sequenced. So this is only like excerpts of the book, right? But now there's almost 3 million genomes available. If you think about the library that would be needed to be built to house all of those books, there's a lot. It would take a lot of infrastructure. And that's what we're seeing with genomic DNA right now, where we have so much data that it's very hard to store it. So here's the Wellcome Genome Campus, where I used to work. So this is where we used to do the research. This is where we used to eat our lunch. And this building here is a giant super cooled warehouse where we can store all of this genomic data. And when I was there, at least, the genome data was the second biggest user of information or data after the Large Hadron Colliders in Europe. So it's a huge problem right now that we're generating so much sequencing data. But all of this sequencing data is really important, and we can use it for really important information. So going back to our microbes, we can use genome sequencing to determine which microbes are good and which ones are bad. So a good bacteria might be something, like a probiotic, like lactobacillus that you have in your gut, like you might find in Yakult. This is what we call a commensal, something that might help you to digest something, for example, and that's not going to make you sick. But the pathogen, like the salmonella that was on my chicken in Mexico, that was bad. But how do we tell them apart, right? They could be very similar. In fact, bugs like E. coli can be either a commensal or a pathogen, and we need to examine the genomes to determine or predict if they're going to be good or bad. So here's an example of something that you might remember from 5 years ago. We were deep in an outbreak of SARS-CoV-2, and the other cool thing that this sort of genome sequencing allows us to do is arrange similar sequences together, what we call clustering, to allow us to look at different types of strains or whatever. So in this case, this is a SARS-CoV-2 tree. Just so each of these lines, there's heaps and heaps of lines here. These are genomes, and basically you can say, remember the Delta variants and how nasty they were? If you're in this red section, you're going to be really sick. If you're in the green section over here, which wasn't such a bad variant, then you're going to be fine. So we can determine based on the genome sequences how, we can at least predict your symptoms and how sick you're going to get. So the other thing, like what I've used genomics for a lot in my research has been predicting antibiotic resistance. So here's an example of a study I did in collaboration with Vanessa Wong and others, basically looking at a large data set of global Salmonella typhi, my old friend, Salmonella typhi strains from across the world. And we had them grouped together in different, what we call strain types, or similar strains that looked quite similar. And what I did was look at all of the antibiotic resistance genes that meant that they wouldn't respond to antibiotics. And so we could track which kinds of strains wouldn't be easily treated that were going to cause those problems. The other thing that we can use genomics for in hospitals, so this is like a large global study, but we can look within hospitals. So for example, this is a high resolution study of a pathogen outbreak in a neonatal ward. This was in Nepal. This is in collaboration with the Oxford University Clinical Research Unit in Vietnam. And we basically sequenced these really nasty infections in these babies in a hospital, and we did environmental sampling. So this means like you start sampling the hospital wards, walls, and the medical devices, and even the nurses and things like that. And we found that this particular outbreak was coming from a nasty dripping pipe that had bacteria in it and was seeding it into the babies. So without doing this kind of tracking, how would you know that that's related to something over here? So the genomic data really gives us power to stop outbreaks. So this kind of work took me all over the world. So that's a tip. If you ever want to go across the world, you can work on infectious disease because it's a global problem. So as Grant mentioned, after I was in Cambridge, I worked in Malawi in sub-Saharan Africa for the Malawi Liverpool Wellcome Trust Clinical Research Program. Here's my team. Here's the Queen Elizabeth Hospital that I worked in, that was 5,000 beds. Here's the research centre that was attached to the hospital where we did our lab work. And this is the Liverpool School of Tropical Medicine that I was employed by, but I didn't work there. So basically when I got there, they were working on, there's huge problems with tuberculosis, HIV, malaria, and so there was a lot of research in those areas. But I was like, What about the hospital-acquired infections? This means that when you go to a hospital, particularly, hospitals, they use a lot of antibiotics, they have really harsh environments. So you can actually, I don't want to scare anyone, but you can actually pick up some pretty nasty infections from being in the hospital. So I was asking about that, no one had checked it. So when I got there, I noticed that when I looked for those hospital-associated infections, that they were on the increase. And I don't know if this is because there was more movement throughout the hospital or if they'd just been seeded and now they were getting picked up more and more. So I started that research program, which is ongoing. And this is an example of one of the studies that we did where we did this genome sequencing and made this pretty tree to show that which strains were really, really nasty in the hospital. And we found that there were certain strain types, this is of Klebsiella pneumoniae, the one that gives you pneumonia, that had a really awful mortality rate in babies in the neonatal ward. And it was completely untreatable by any of the local antibiotics. So it was a death sentence if you got this thing. So you can see how important it is to track particular types of strains, but other ones that just pop up and then go away, they're not as, you don't have to sound the alarm as much. No, I'm so sorry, I forgot to mention that. So 90% of the, by far majority of all of these hospital-acquired infections are caused by one of 6 bacterial species, which the acronym is ESCAPE: Enterococcus, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter. Yeah, so I checked for those ones. That's how I knew how to pick those ones up. Yeah, thank you. All right. So that's sort of a really big global view of how you can use genomics to look at infections, outbreaks, global tracking, those kinds of things. But the other thing you can do is zoom way, way in a lot of detail. And I did a lot of this during my PhD and still are doing it, just out of interest. So antibiotic resistance genes are not just randomly distributed throughout the genome. We're not just sort of blindly expecting one here and one here. What they tend to do is cluster together in these, what we call multi-drug resistant regions. So the resistance genes, the things that give resistance to antibiotics, are colored. And what you'll notice is there's 9 genes that are clustered together in a small section of the genome. This is a very small, this would be like 0.0001% of the whole genome, very small. And the scary thing about this is the whole thing is mobile. So this thing can pick up and move between strains. This would be like if I could pass you genes as an adult to an adult, just like this. And say I wanted to pass you genes for curly hair, I could give it to you just by being near you, and then your hair would spring up immediately. But in this case, you can get resistance to 9 antibiotics, just like that. And the way that works are these things called plasmids, which can move across cells. So here we have our antibiotic resistance cell in red, and it passes this thing to its adjacent cell that's sensitive, and it makes it resistant as soon as it enters the cell. And so, so this is an example of a really nasty thing. And to make matters worse, this is actually from an animal isolate, an Australian animal isolate that's a food-producing animal. So you can see why it's really important that we don't use antibiotics in animal feed, because this really encourages the growth of these things, that as soon as you're like near animals like this, or you eat them, or with water sources, there's a whole bunch of ways you can get them transmitted. They can jump to humans really easily, and they can even live in your commensals, in your microbiome. So you have to be really, really careful about preventing this kind of thing from getting into animals. So that's a lot of the stuff I was doing using genomics, and why I think genomes and genes are really important to be tracking and understanding what they're doing, particularly in microbes. But now a lot of my research focuses on understanding what microbial genes do. So what does this mean? So it's really important to understand just generally what genes do. So this is an excerpt from the old the old administration in the US. I don't think that they have this anymore, but I think it's important to point out that it is a really important goal to, in this case, they were looking to sequence 1 million new microbial species and understand what 80% of those newly discovered genes do, right? And this is because there's a whole bunch of, like, as I said, all of these superpowers just waiting to be discovered. And the best way to do that is by looking through these new genes. So you might think, oh, well, yeah, it might be a little bit lofty, but it's doable. Let me tell you, who here has heard of E. coli? Like E. coli? Everyone, thank you. So we've been studying that for decades and decades, billions of research funding going into this one little bacteria. We still don't know what a quarter of its genes do. 1/4, we don't know. That's because it's really hard to find out what genes do. So how do we find out what genes do? Well, there's sort of a hierarchy of the sorts of things that we can do. At the top, the gold standard is experimentation. This, I wish I need my explaining hands for this. This is if you have a wild type cell, something that's occurring in nature. Can you guys still hear me? Okay. A wild type cell that's occurring in nature, and you want to examine what one gene does, right? So you take that cell and then you disrupt or knock out one gene in that cell, and you compare it to the wild type undisrupted, right? But how do you know what to test? You can do millions and millions of assays until you find something that's different between that one, what we call a mutant, that's lacking that gene, right? So this means that experimentation is great, but like professors can spend their entire career working out the function of one gene. Then on the other side, we have sequence homology. This is where we get our tons and tons of sequencing data that I told you that's just piling up there. And we say, okay, this gene that's been sequenced looks about 40%, maybe 50% similar to this other gene that this professor has spent 20 years working out what it does. So I'm just gonna say that it does the same thing. But often, that's not the case, because especially if it's in a different organism or something like that, you can't transfer function in that way. You can't infer that. So it's not a great method, but it is quick and cheap. So a lot of the gene function is done this way, by transferring knowledge. Where I work, that in this sort of in-between section that I think is just right, is called functional genomics. And this is what I've been working on for the last 15-ish years, because I think it's a really important thing to be able to assign gene function that has a little bit of experimentation, a little bit of high throughput, to do this on a large scale. So how do I do this? My favorite method to do this is a thing called TRADIS. Now, what is TRADIS? It's not this. It stands for Transposon-Directed Insertion Site Sequencing, which is a bit of a mouthful, that's why I say TRADIS. If you're interested in sort of reading up on this is a review I wrote that sort of summarizes all the cool uses of this method. Over the last years, there's been tons and tons of different things done with it. But really, all it's doing is taking that whole genome sequencing data and combining it with large-scale mutagenesis. So instead of disrupting one gene at a time, you can disrupt all of the genes, millions of genes at once, and you combine that to be able to determine the function of each gene in a bacterial genome for their involvement in any condition. So that's a really complicated concept, even for microbiologists. So I'm going to give you a little bit of an analogy that I like to use. Okay, so imagine that I have a fleet of 1 million cars, right? And I want to know what car parts do. Like, I don't know. I'm a girl. No, just joking. I don't know what car parts do. I'm a scientist. So imagine I get blindfolded and then I smash up each of these 1 million cars randomly, one smash at a time. So what happens is I get a car that has a smash in the roof, a car that has a smash in the headlights, and a car that has a smash in the wheels. So let's see how they drive. The one with the disrupted roof can drive just fine. The one with the disrupted headlight can drive just fine. The one with the disrupted tire can't get very far, right? So what I can conclude, now I know that wheels are needed for driving, but headlights and roof aren't needed for driving. Okay, but what if I change one thing? So now it's nighttime, and we do the same thing. I smash up the cars, and I get them to run the race. I get them to start driving. So we have the roof, the headlight, and the wheel. So the roof still can drive just fine. Now, the headlight suddenly becomes important. That car's not going to get very far. You can't drive home on one headlight. The wheel still remains important for driving. So we can conclude that wheels and headlights are important for night driving, but the roof is not. And you can imagine you can apply this for any condition. So now it's raining, what becomes important? The roof, okay? So you can see how we could apply this to bacteria in a way that we can start to ask pointed questions around what genes or parts of the bacteria are needed for resistance to antimicrobials. Infection, like how do they come and become pathogenic to an animal? Or how do they survive stress conditions? These are the kinds of things that we ask. And we also combine TRIDIS with other multi-omics technologies like proteomics or glycomics to fully understand what's happening in the cell. So as I said, my group, we study a lot of antibiotic resistance, which I'll talk about in a second, a little bit more about how we apply that. Plastic waste, I'll also mention how we use these sorts of functional genomics to study plastic waste. We also can use it to look at microbiome and in our model organisms. We can look at how bacteria deal with stressful conditions. So for example, my favorite bacteria, Acinetobacter baumannii, this is one of these nasty escape pathogens. It can live on hospital walls for months at a time, even a year, and it's even more pathogenic than when it started. So they're really good at what we call desiccation, and we can understand how that can be, and we can make new antibiotics that specifically target those kinds of things. We're also really interested in microbial cooperation and co-infection. My student Jasmine's here, she can talk to her, that's one of her projects. We're finding more and more that bacteria help each other to co-infect in hospitals as well. So I'll just touch on a few of these points. So let's look at, so we know that antibiotic resistance is a problem. Are there any solutions on the horizon? Sadly, no. There's thousands of anti-cancer drugs in them, sort of in development, but there's very few antibiotics. And it's been a long time. It's been decades since we've had a really good breakthrough of new antibiotics. And a lot of this is because the low-hanging fruit of known targets. So when you're looking for a target for a new antibiotic, you want something that'll kill the bacteria, but you don't want them to hurt the human cells, right? So one thing that we can do is sort of look at new antibiotics' mechanism of killing, because maybe it'll be a new mechanism. So this is an example of a study I did in collaboration with the University of Queensland. They found there was a new antibiotic hiding in a lugworm, which is a beach worm. It tells you why you should do bioprospecting. But yeah, this antibiotic was called arenicin 3. It's a polypeptide antibiotic, and it was really potent against those nasty escape pathogens and also E. coli. So they were really excited. But because it came from a worm, they had no idea how it was working. So this is where we can use TRIDIS, right? So you take the TRIDIS, what we call the libraries, and we can grow them with the antibiotic and see which genes are needed to survive in that antibiotic, and we can work backwards to see what the antibiotic targets. So we worked out that it's definitely going for the inner and outer membrane, and then once it's inside the cell, it actually slows down how they replicate. So that was the first application of using this kind of technology to study the mechanism of a drug. The other thing that we've done is applied this technology to understanding how biocides work. What are biocides? The WHO defines them as essential medicines that help to control infections in hospitals, but also you'd see these all throughout your home because we use them for cleaning. Things like bleach, ethanol, some disinfectants, and detergents. But we don't really know a lot about how they work. We know they're pretty good, pretty potent, but we don't know how they work. So this was a study that we did. We used that really nasty bacteria, Acinetobacter baumannii, and showed how all of these biocides killed that bacteria. And we found that it actually had a completely unexpected mechanism of action, which was de-energizing the membrane of the bacteria. So it was kind of like disempowering it. And then the last thing I wanted to touch on is that Chartis is also, or any sort of functional genomics is a really good way of finding new vaccine targets. So we're kind of realizing that vaccines are really important, right? Especially again after the COVID outbreak, that stemmed it very well. So in this case, this was an animal disease called strangles, which sadly is caused by Streptococcus equi. It gets into the lymph nodes of the horse and it strangles them, literally. And it's the biggest killer. It's really awful. The vaccine, when I started this study, the current vaccine wasn't very effective, had really bad side effects. By that, I mean the actual vaccine caused the disease in quite a few percent, like, 3 to 4% of the horses, which is crazy. So we needed a better vaccine. So we put our Chartis libraries of Streptococcus equi through the horses so we could start to see which genes are important for infections and which ones could we use to identify new vaccine targets. And this worked really well, and we got a vaccine called Strangvac, which is now on the market in Europe and has been for a couple of years. So it's a really good way of finding new targets. So I hope that I've convinced you that Chartis and functional genomics is a really good way of finding out what genes do, and I hope you can understand why finding gene function is really important, especially in the medical sphere. But what other major problems can we tackle? Well, the other thing that my group research is the problem of plastic waste. So we all know that plastic is a mounting problem. We have a lot of plastic waste generated each year, and there's not great disposal routes. So we put a lot into landfill. Even in Australia, this number's even higher. And some countries incinerate a lot of plastic, which has a lot of nasty byproducts. And the downcycling, I think that's the worst. If you ever look at those plastic park benches that say made from recycled whatever, it just means that it's just going to be there forever. At the end of time, that plastic bench is going to stay there because it can't be recycled again. It's so solid that it can't ever be recycled. So the two plastics that we focus on are largely polyurethane. These are probably in the seats that you're sitting in right now, that foam, as well as lacquers and paints and things like that. And then LDPE, which is really a very tough plastic that, it's just like long chains of carbons. And these are in single-use plastic bags. They're really flexible. So we need a better solution to recycle plastics. And obviously this touches on a number of sustainable development goals. Microplastics are entering our system as humans. We're becoming more sterile. They're getting into our brains. We're eating a credit card of plastic every week. But also there's going to be more plastic than fish in the ocean in 2050. Like microplastics and chunks of plastics, plastic islands are out in the ocean. It's a mess. We really need a better solution. So now let's talk about these little caterpillars. Some of you guys have seen them eating plastics. These guys are called Galleria melanella. Now these are the greater wax moth larvae, and they're really, really, really hungry, right? They're voracious eaters. And if you put about 100 of these caterpillars on a plastic bag, about 13% will be gone overnight. And you can see this, actually, this is the kind of plastic bag we're talking about. This is an overnight with 4 caterpillars. So they're really efficient, and very few things can do this. If you were to leave this plastic bag in the landfill, it would take hundreds and hundreds of years to degrade, and this would go into microplastics. So can't we just put tons of caterpillars on heaps of plastic? Unfortunately, these guys can't be released into the wild because they're pathogens of beehives. They get into beehives and they completely destroy the hive. So we definitely don't want to release them. But the reason that they live in beehives is the reason that they're so good at eating plastic, because they've evolved for millions of years to eat beeswax, which is structurally similar to LDPE, for example. So it's kind of their blessing and their curse. Maybe they can turn around, do some good deeds for us. So how can we harness the caterpillar's powers? Again, clue is in the title of the talk. We can understand what genes are driving this plastic degrading ability, and we could then take those genes and put them into safe workhorse microbes. And we do this via synthetic biology. So what is synthetic biology? It basically is a field of science that treats genes as modular, standardized parts that can be constructed into biological machines that make something that's useful for us. And if you think about them like Lego blocks, the more genes we can discover, the more variety of Lego blocks, you start getting all those expansion packs, you start making really, really cool things. So we want to know more about what genes do to have more Lego blocks that we can use to build cool synthetic microbes. So this is sort of the philosophy underpinning synthetic biology, that basically we can design constructs, build these synthetic microbes that don't exist in nature. We test them, are they good for the purpose, and can we make them better? So we learn from how we can optimize them and actually make them efficient for application and industry. And so obviously a lot of the work that I do in gene discovery is in the sort of initial design phase. Now, synthetic biology isn't something that's just a theory or a pipe dream. It's actually a multi-billion-dollar industry that is used. You'd be surprised how many things that you use are produced using synthetic biology. So for example, insulin is made using microbes. Instead of killing a whole bunch of pigs and taking the insulin from their pancreases, which isn't very ethical or nice, you can just take a microbe, get the insulin producing genes, pop them in there, and there you go. The other thing that you might know is like lab meats, where they took the hemoglobin gene, which is really red, put it into soy, and so you have nice, realistic, bleeding style plant-based meats, right? So we are actually here at Macquarie a bit of a leader in synthetic biology. We have this ARC Centre of Excellence, which is Australia's sort of biggest funding platform, which has $35 million, 21 industry partners, and we've raised $210 million of capital across 10 startups. And we do heaps of cool stuff with strain construction, protein production, blah, blah, blah. We also have on site the Australian Genome Foundry, and this one has huge it's an infrastructure grant facility that's sort of supported initially by the New South Wales government to allow us to be able to really upscale all this stuff. So instead of having single people pipetting one at a time, you have robots that are doing thousands of plates at once. And this means we're making things more efficient and we can screen way more things. The other thing that we have on site is the Galeria Research Facility, which I helped to co-found and run with Hui Din, who's injecting some Galeria there. And this is basically using the Galeria not only to study plastic as we're doing here, but it's a fantastic ethical animal model alternative to mice when you're studying drug toxicity, efficacy, but also infection model. So we use it for lots of different stuff. And lastly, we have the RNA facility, which is being built on site right now with Myeloid and the New South Wales government. So let's just think about how we can use synthetic biology to break down plastic. So there's already a precedent set. About 10 years ago, in 2016, there was a microbe called Eissechiensis that was isolated from a rubbish tip, or just outside a recycling plant in Tokyo. And they found that this guy was really good at degrading PET plastic, which is the kind of plastic you find in these sorts of plastic bottles. And the way that they do this is they produce these enzymes, which are like little Pac-Man that chew up everything, called petases. And basically, they cleave these certain bonds within the PET plastic, and break it down into byproducts that are safe. So this guy, you can imagine, you can take, once you find out what the genes are that are driving this reaction, you can put this into a safe workhorse microbe and then start to produce that. And you can recycle plastic bottles that way. What do we know about other plastics? So for example, LDPE or polyethylene, like the single-use plastic bags. Yes?
Is the PETAs created in a similar method to how insulin is produced in bacteria?
So this one's a naturally occurring one, the Isakiensis. So the insulin producing one, that's completely synthetic. Like we took the enzymes from the pigs and then we put them into a microbe, but this one here is naturally occurring at the moment. So we just found it in a rubbish tip.
Could it be put into some kind of synthetic bacteria?
Yeah. Oh yeah. We're trying. Well, this sort of stuff is still all ongoing. This thing, I don't know if it's actually published, but there is a company that does this in a pretty, like they recycled all of the plastic bottles at the Australian Open one year using these enzymes only. So it can be done at scale. Yeah, maybe it can. Maybe. Exactly. Yeah. Exactly. Yeah. So we want to know how they're doing all this stuff, right? So we can start to. So this one we know how they produce these things, but with the other plastics like the LDPE, like the plastic bags, we don't know how they're breaking them down, right? There's a handful of examples of some of the Pac-Man enzymes that can break them down into smaller bits, and then there's some evidence showing that the microbes can then take them into the cell and use them like energy, but there's not a lot of detail on it. So what we do in our lab is we train the Galeria to eat plastics. So these guys, actually these ones weren't trained. This is just how they are normally. We can make them even more efficient. And then we can bust them open, and often we take the microbes from their gut. We think their microbiome is really important for breaking down plastic. And then we identify the building blocks or the genes that are driving this plastic degrading activity. And then we put them into the safe workhorse microbes, like the synthetic microbes, right? Like the yeast or whatever we want to use. So you can talk to some of my team about this. And we're at various stages, but I thought I'd give you one case study of a paper of my student Fiona Facey that's sitting there that we just got out today, literally today. So this is fresh off the press. So we fed in this case, we fed the caterpillars some plastic bags. We homogenized them. This means we just sort of mushed them up into a base, and we plated them onto agar plates. And we looked at their ability to break down the LDPE. So this is just taking like a film of that type of plastic, like the single-use plastic bag plastics, and then we look for evidence of what we call like pitting and cracking. So you can see they're starting to break these down. But then we thought, hey, you know what's more important is like multi-layer plastics, right? Often what people don't realize is plastics that occur like these. This is different. This lid is a different type of plastic to the bottle. There's a lot of different types of plastics that you see in packaging and you don't realize they're in layers. Crisp packets like this have at least 3 different types of plastics in different layers. And so it's really hard to recycle them. But these caterpillars, they can eat them. So they can eat them without any problems. The other thing to think about is like mixed waste streams. Like when you're recycling plastics, you kind of just put them all in together. But often it's really hard to separate them. So if we can get solutions like microbes that can break down multiple types of plastic at once, that would be great. Anyway, so we decided to test another type of plastic, polyurethane, like the ones that you're sitting on. And we found that one of these isolates could actually break down not only LDPE, but also PU. And here we have the plastic sitting in the plate, and you can see a clearance zone around it. So we next sequenced the genome of this multi-talented bug, and we found that it was something called Bacillus subtilis, which has been associated with plastic degradation before. We found a number of genes that had been previously associated with LDPE and PU degradation, and we're currently working on, this is photo from my student Sam, that using a whole bunch of different cool microbes to put these genes into and then assess their plastic degrading ability. So here we have one that's causing a lot of plastic clearance here, and this one too, because I mentioned sort of baker's yeast and things like this, but we might have better hosts that we can use for our synthetic microbes. Yeah. Cool, so that's pretty much it. I was just going to wrap up, and I hope I've convinced you that fundamental research is really important to drive these kinds of cool applications. Functional genomics and finding out gene function is really critical to unlocking these really cool powers that microbes have. Synthetic biology is a great way to tap into really, to harness the powers of the microbes, but also there's innovations, like within the Center of Excellence, there's a handful of people here tonight, and everyone does really cool research. So some of the cool other things that we're applying synthetic biology to are like biofuel production, methane reduction, waste recycling, production of valuable products like in cosmetics or pharmaceuticals, and animal-free alternatives, for example, like the meat example I gave you. And if any of this resonates with you, the future of Symbio lies with you guys. And so if anyone really wants to get involved in Symbio stuff, we have not only just sort of the third year subjects on Symbio, so if anyone's interested, like I teach into those subjects as well, but the iGEM team and the Oz Symbio Challenge are national and international synthetic biology competitions that allow students to do really cool Symbio. Absolutely amazing stuff comes out of those competitions. So come find me, or send an email or whatever if you're interested in joining one of those things. Cool. So I would just like to, again, thank the organizers for giving me the opportunity. I'd really love to thank my team, particularly the ones that came tonight, so Jasmine, Fiona, and Brody. And just everyone is just so amazing, like all of this research on the ground, a lot of that is from them. My amazing collaborators, and all of my funding, obviously. Wouldn't get anything done if I didn't have funding. And you for your attention. Thank you. Questions?
Wow. Thank you so much, Amy. I've learned two quite interesting things tonight. One is that you have a favourite bacteria, Acinetobacter baumannii. I'm not a microbiologist. And that you are excited about finding things in rubbish tips.
Yes.
But also something else you mentioned about being in hospital and you come away with all these nasty infections and so on. I just want to assure everyone in the room, Macquarie University Hospital, absolutely fine. Just a word from our sponsors.
All of the other ones.
All the other ones. I'd like to open the floor for questions. Anyone have any questions of Amy? Please just raise your hand and we'll have the microphones. Oh, over to the left.
Thank you for your very enlightened talk. I'm just curious, with the new synthetic compounds you're making, is there any danger to humans working with them? Do you take precautions at all when creating these things?
Yeah, so actually we use the Galeria as a high throughput screening method for toxicity. So when we're dealing with new compounds, we first test them on cell lines, so the sort of most simple of like human cells. And then next we put them into the Galeria, so we can put them into an animal, and then we'll also test them in mice, but down the line, because we want to try and minimize that. But yeah, 100%. And we also have very schmick new, what we call physical containment level 2 labs. So Fiona, who's worked in the Galeria facility, can tell you have to wear a hairnet, booties. It's a very, very strict safety procedure. So we're very careful with everything in the lab. Yeah.
Mine's a broad question. I'm a child of the 80s, and I just wondered, I have a very strong immune system, and is that a factor of my environment? So I grew up before food safety was a big thing, germaphobes, etc. So left out in the garden to, eat dirt and grass and build up a good resistance? Or is it related to genetics? Some people have a stronger resistance in general. And do you do the reverse testing? So you test the microbes and the effect and break down all the cars. Do you do the opposite? And are there people that have a higher resistance and then do the opposite or not?
Yeah, so the thing that is the best at, so what you're talking about is really microbiome. I think that's really the thing that affects your immune response and all that kind of thing the best. The best way to get a healthy microbiome is spend time around animals, particularly farm animals are very good. Obviously not if they have really nasty resistance, but yeah, even sharing the house. They've shown that people that share houses with animals, for example, tend to have less allergies and things like that. So you can, and actually the other funny thing is it's not related to genetics. If you and your sister or whoever is living in different households, even though you had the same upbringing in the environment and half genetics shared, it's actually people within your household that you'll share your microbiome most with. So it's about co-inhabitation, including the animals. Yeah. Click it on. Just.
Hello. Thanks for the talk. When you say you train the Galeria caterpillars to eat plastic more efficiently, how does that work?
Yeah, so we, okay, so when we're using them in the lab, for example, for infection studies, we'll give them a diet that's more like, we actually pampered them a little bit. We give them baby food and honey. It smells amazing. So because we don't need them to be, we want them to be growing as quickly as possible, and we want them to be as healthy as possible. When they eat the bees wax, like in nature, it's like giving someone a really, like a fast food diet versus, you know, their natural diet. It just makes them grow a little slower and that kind of thing. So what we do when we want them to eat the plastic really fast is we give them beeswax to start, and then we add more and more plastic into the diet. So it's about just sort of, if you take the ones that have been trained just on the baby food and then you get them straight on the plastic, they can be a bit grumbly. They're like, hey, where's my baby food? Yeah. A couple of the people in my lab are definitely caterpillar whisperers. They're really good at getting them to work for us.
Getting my exercise in.
Yeah.
Thank you. First of all, I thoroughly enjoyed your presentation, and it's very fascinating to know more about the mechanisms, techniques. But my question kind of does extend a little beyond that, on something that you mentioned briefly. It was on the transferability of, I believe, pathogens in animals, and you mentioned that briefly. How does that actually work? Is that a sort of like a proximity thing? Is it a skin-to-skin contact which sort of comes to that, or I was just quite interested.
So, well, yeah. Look, I usually do a whole lecture on this. It's basically that it's a global problem, and it's what we call a One Health solution. So when you are thinking about antibiotics, like an antibiotic resistance and being a conscious user of antibiotics, right? You think like, oh, okay, I need to finish my course of antibiotics when I have them, and I won't go to the doctor if I have a flu, right? But this is a very like human-centric view of antibiotics, whereas in reality, 80% of antibiotics are used in agriculture and veterinary medicine, and actually human usage is actually a small percentage. And then how does that cross over? Like, how do we get those nasty microbes? A whole bunch of different ways. So I just have a grant in at the moment on looking at, I'm so sorry, I'm going to ruin everything for everyone. Supermarket meats. You can see really nasty resistant pathogens in supermarket meats. You can see them in runoff. So a lot of it's coming from, like, say, waterways as well. So if you have farmland, so you have the animals and they poo in all the soil, and then you have a big, like, there's a lot of flooding at the moment. All of that washes into waterways, and then what happens? We drink it. A lot of the sterilization that Sydney Water doesn't actually get rid of, I don't want to freak anyone out, but it doesn't get rid of like genetic material, does it? So you can, it's not, I don't want to freak anyone out, but you can get it through just the waterways and just everywhere. No, it's that's why we call it like a One Health solution, because it's everything. It's also so it's eating the meats, it's the runoff from the water, but it is also close contact. You know, like with the SARS-CoV-2, like that was suspected to be, you know, people that are close to animals in markets and that kind of thing. So that obviously happens on farms as well. But it's not probably the primary thing, I don't think.
Any more questions?
One more.
So if you make these microbes that can degrade plastic, can I go and infect Woolworths so that eventually all the PET bottles explode?
Pretty much. I mean, we do work with, well, we try to work with Woolworths and stuff like that, because like you saw the red cycle. I don't know if anyone remembers. Remember when you had to take your soft plastics and put them in the red cycle thing, and then they realized it was a big scam because there's no way to properly recycle those things. So we're trying to give solutions to do that. Yeah, so we do want to get our hands on their waste, yeah, so we can play with it. You're like, come on this side. Could you give her a round?
Thank you for the lovely talk. I just wanted to clarify. So, like, the TRADIS is like a trial and error kind of process, right, of finding the specific gene that you guys want. I just wanted to ask, how long would it take for that entire process of, finding the specific gene? Like, is there an approximate time, or is it like until you really find it?
So it's actually really fast once you have it set up. So we get answers back in, I don't know, 2 weeks, something like that. So say you wanted to find out, okay, I've got a new antibiotic, I want to see what genes are involved in resistance or survival in this antibiotic. You do the assay, that takes a couple of days. You extract the DNA, that takes a day. And then you send it off for sequencing, that takes a week, or a couple of weeks maybe, and then you get the data back. So it's pretty quick. What takes time is making that million mutant library, like making the fleet of cars that smashed up. That takes a little bit of time, and that's variable on the organism. So it can take anywhere from, I made one like overnight once. No, it usually takes probably a couple of weeks to a couple of months. So maybe up to 6 months, depending on the organism.
We have time for one more question.
Yeah, thanks. An off-the-wall question, Amy. Complementary to the way you approach antibiotics, I've heard antibiotics described as by metaphor, sort of a lock and key mechanism. You can look at a bug and sequence its shape or something, look at a protein, and then maybe find a key that fits it and disables it in a particular way. So you can do the whole thing almost at a chemical level. You're clearly not doing that. You're approaching a different mechanism. How do the two approaches compare? How are these two systems compatible? How does what you do sit with what the proteomics people do to find antibiotics that way?
Well, if I understand it correctly, you mean like, there's sort of different ways of doing drug discovery. You have like cell-based drug discovery where you're like, okay, I want to target this, let's do a giant chemical screen and see which kills it. And then you have target-based drug discovery where you have a target and you're like, this is a really good target, I know this is going to kill the bacteria. And then you like, look, okay, so this is to do with protein folding, I need to find a chaperone that can fold that protein correctly. How do I find that? And you can look through different chemical libraries or whatever. So we do that sort of thing as well. So that's what I'm kind of saying about the drug target. So we often find targets that then we might work with chemists or synthetic biologists or whoever to make the compounds to disrupt that target. But the other thing I'm not sure if you're talking about is we've only just recently, there's probably the most cool emerging new tech that's coming out of synthetic biology is that we can design proteins. So we can design proteins that we want to do a certain function. So say like, you know, you want something to fit in a certain spot in the cell or whatever, we can do that, and that's literally just starting in the last year or so, right? Yeah, like, so that's a really cool new emerging tech, but I think there's only like a handful of publications in that area. But that's a really cool area that I'd love to move in at some point.
All right, might wrap it up there, but please feel free to come and have a chat to Amy after the conclusion. So, I don't know about you, but I have learnt a lot tonight. In particular, I'm very much looking forward to the day that we see Woolworths, industrial espionage at Woolworths with a horde of caterpillars moving through the aisles. I think that's going to be a great day. Until that time comes, please join me in thanking Amy for an absolutely wonderful presentation tonight. Thank you all so much for coming. Please travel safely home, and also remember our last Discover lecture for the year will be by Professor Rick Shine. Rick is a superstar in all things frog, particularly managing cane toads in Australia. QR code, get yourself registered, get along, 18th of November. It's going to be a fantastic presentation, just as it was tonight. Thank you all very much. Have a great evening. Thanks for coming.